In this presentation, I'll talk a little bit about first and second messengers, as well as about AMPA receptor trafficking. First messengers carry information between neurons. Neurotransmitters, such as glutamate, frequently act as first messengers. Second messengers can carry the signal into the neuron. So it's important to remember that neurotransmitters like glutamate don't actually enter the postsynaptic neuron. They bind to receptors on that neuron, and those receptors can then allow second messengers to either enter the cell, which I'll explain in a minute, or they can activate other second messengers that are already in the postsynaptic cell. Once they are mobilized, second messengers can passively diffuse throughout the postsynaptic cell, where they can interact with proteins. They can greatly amplify the effect of the first messenger. Some examples of second messengers are calcium, cyclic AMP or cyclic adenosine monophosphate, and some membrane lipids can be converted into second messengers. Examples of these are diglycerol or DAG, and inositol triphosphate, or IP3, which we'll talk about later in the semester. In this discussion, we'll mostly just talk about calcium as a second messenger. So calcium can enter through the NMDA receptor's ion channel and act as a second messenger in the postsynaptic cell. So here you see graphically calcium entering through activated NMDA receptors, entering the postsynaptic cell, and there they can initiate biochemical interactions. The purpose of this is primarily to modify proteins that already exist in the dendrites. This is an example of post-translational processing. This just means that the function or structure of the protein is altered. This little animation shows first messengers and second messengers. So you can see a presynaptic cell on top with some neurotransmitter packaged in the vesicles. Those are the little blue dots. And on the postsynaptic cell, you can see some receptors that will allow calcium, which are the orange dots, to enter the cell. So here you see an action potential invading the neuron. Glutamate binds to receptors, causes calcium to come in, and they can rapidly mobilize and carry that signal that's arriving from the glutamate into the postsynaptic cell. So here, glutamate is the first messenger, calcium is the second messenger. Second messengers typically target two specific types of proteins, and those are types of enzymes that can either phosphorylate or dephosphorylate other proteins. So protein kinases can phosphorylate or attach a phosphate to proteins and alter their function, and phosphatases are enzymes that can remove phosphates from proteins. So binding of second messengers to these kinases and phosphatases can activate them and allow them to do their job. So here's another animation that looks a lot like the one you just saw. So glutamate, the first messenger, can bind to receptors on the postsynaptic cell, activate those receptors, and allow calcium, the second messenger, to enter the cell. And once calcium is in the cell, it can activate the kinase that you see down there on the bottom and cause it to change its structure and thus become functional. So there's glutamate binding, calcium can come in and activate that kinase. So those are first messengers and second messengers, and how they can work together to activate kinases in the postsynaptic cell. So the second topic of this brief presentation will be the trafficking of AMPA receptors. So remember that a primary mechanism of the expression of LTP involves increasing the number of AMPA receptors via trafficking. So AMPA receptors are trafficked. They start out attached to vesicles just within the postsynaptic dendrite. They can move closer to the postsynaptic density and then finally into the postsynaptic density. And those changes are caused by phosphorylation of the GLU-R1 subunit of the AMPA receptor by different types of kinases. And there's 3 basic steps that you see below. So first, those vesicles containing AMPA receptors are mobilized. They're mobilized to move those AMPA receptors closer to the postsynaptic density, or PSD. So that occurs via activation of protein kinase A. Those receptors can then diffuse to the postsynaptic density, so they're then actually in the synapse. And that change is produced by activation of protein kinase C. So step 3 is that some of those receptors will then become trapped and immobilized within the postsynaptic density. And that change occurs via activation of CAM kinase 2. So the trafficking occurs in 2 stages. On the left, you see stage 1, where those AMPA receptors that start out in vesicles can move closer to the PSD. They're not quite in the PSD yet, but they move into the cell membrane in the extrasynaptic region. So that's the mobilization step. So extrasynaptic just means they're in the membrane, but they're not right in the synapse yet. In stage 2, and back to stage 1, that, remember, is caused by protein kinase A, or PKA, activation via phosphorylation. In stage 2, calcium coming in through NMDA receptors activates protein kinase C, or PKC, and that causes those receptors to move or diffuse to the postsynaptic density. So now they're in the postsynaptic density. If there's more AMPA receptors there, then the same amount of glutamate can cause a greater amount of depolarization in the postsynaptic cell. And so that's how not the induction, but the expression of LTP is dependent upon all these extra AMPA receptors in the postsynaptic density. And so here's another little animation. Glutamate will come in, bind to, this one skips the glutamate step. So it starts with movement via PKA to the postsynaptic density. Protein kinase C is then activated via glutamate receptor calcium entry, and that protein kinase C then causes those receptors to move into the postsynaptic density. Once in the synapse, there's a 3rd step involved in trafficking, and this is that those AMPA receptors can become trapped in the postsynaptic density by CAM kinase 2. So CAM kinase 2, remember, is a holoenzyme, so those are the round purple things that you see. And one of their jobs is to hold together the F-actin filaments. So remember, F-actin is the form of actin that looks like a strand, so it's a bunch of subunits all stuck together. That provides support to the postsynaptic dendrite, and it can also trap or hold those AMPA receptors in the synapse so they can't diffuse back out. And CAM kinase 2 has another job. It can also increase the channel conductance of AMPA receptors. So this means that they can allow more sodium in, but they can also allow calcium in when they're activated by CAM kinase 2. So these 2 things together mean that there's greater conductance of those AMPA receptors, and the same amount of activation of those receptors by glutamate can result in a greater depolarization of the postsynaptic cell. And that's it for this one. I hope I can get the audio and video together, and I'm going to try and make this into a movie that I can then put up as a movie instead of a PowerPoint slide. Thanks.